Good morning, everyone. My name is Arjena Valls. I'm a student here at St. Thomas University, and my summer internship project is about the molecular cloning and production of the zebrafish chondroitin sulfate proteoglycan neurocan. Now that we understood the role of neurocan in vivo, I'm going to talk about the role of neurocan in vitro. We want to use different approaches to study the neurocan behavior in vitro because it's a more simplified system. So my hypothesis is that the behavioral response of zebrafish brainstem cells to zebrafish neurocan will be similar to that previously observed with the CSPGs, meaning that it's going to be repelled and inhibited. Since the CSPG mix from the chick contains the neurocan. Before proceeding to explain my project, we created a research strategy to set the order of our experiments. First, we are going to clone the zebrafish neurocan complementary DNA. Then we're going to transfect the neurocan into mammalian cells in order to produce the protein. Then we're going to isolate and purify the neurocan protein. And we're going to grow the zebrafish brainstem cells on a neurocan substrate. As I mentioned before, the first step for this project is the molecular cloning of the neurocan. It is important to understand what cloning stands for. It is a process that involves the production of sexually identical individuals. The technique that we use is the recombinant DNA technology, also known as the molecular cloning. This process involves the insertion of foreign DNA fragments into previously cut vector backbones in order to recombine the DNA, therefore its name. We can see this process by this diagram where we have our circular plasmid double-strand vector that is going to be cut by two restriction enzymes, and then we have our DNA of interest, the insert. Then we're going to put them together in a process called ligation. So basically this is a process of cutting and pasting. The steps that we follow for the cloning are digesting the plasmid with two different restriction enzymes, and we're going to do the same with the insert with two different restriction enzymes. Then we're going to phosphatate the vector to prevent the religation. With phosphatase, I mean that we're going to remove the phosphate group from the DNA in order to prevent self-ligation. Then we're going to ligate the insert and the vector together, and we can see this process by this diagram. Here we have our vector backbone. We use a PCMB-MYC vector that expresses the neurocan purification with a MYC affinity tag. The insert preparation, we use a neurocan B. And as you can see here, we use the same restriction enzyme for both, the ECRI1 and the ZOE1. After we ligated them together with the enzyme called DNA ligase, we can see that the size of the PCMB-MYC neurocan is of about 3.8 kilobases. The following steps for the cloning is to transform the bacteria and isolate the plasmid DNA from bacterial culture by the process called mini-prep. Then we have to identify the positive clones and produce more of it by a process called maxi-prep. I'm going to explain in more detail what a mini-prep is. The main purpose of it is to purify the plasmid, and we do this by three different steps. The cell lysis. This process involves the lysis of a bacterial cell, and we're going to separate the proteins and the DNA from the plasmid DNA. After we add the neutralization substance, the proteins and the DNA are not going to be able to renature, meaning that they're going to be separate from the plasmid DNA, which is what we want. the plasmid DNA is going to be later filtered and is going to be eluted. After we identify our positive clones, we're going to proceed with a process called maxi-prep, which basically involves the same procedures as a mini-prep, with the only difference that a maxi-prep produces higher concentration of the plasmid DNA. Here you can see the same steps. We lyse the bacterial cell, we neutralize it, then we filter it, elute it, and it gets purified. To obtain these results, we did a diagnostic gel with two different restriction enzymes, the E. coli and the XBAR1. We obtained 7 positive clones, as you can see here, clone 3, 4, 5, 8, 9, 10, and 15. We know that they are positive clones because of the double strand, double digest, and because the bands are at the expected base pair in comparison to our plasmid map. For our purposes, we use the clone 9 because it had the higher concentration. We later did another diagnostic gel from the maxi-prep in order to confirm that the clone 9 had the vector, the MIG vector. We obtained positive results, and we can see this because the bands are at the expected base pair in comparison to our plasmid DNA, our plasmid map. Also, the maxi-prep yielded a concentration of 625 nanograms. After obtaining these results, we sent the clone for sequencing, and it was confirmed. The MIG was upstream to the Neurocan in-frame. After we have these results, we can then proceed with the transfection. What transfection means is to introduce foreign DNA into a eukaryotic cell. This is usually done by creating an electric field for the cell in order to increase the permeability of it. We did this by using the AMAXA electroporator with 293T mammalian cells. The steps that we follow for the transfection are introducing the plasmid DNA into the mammalian cells with the AMAXA electroporator. Then we isolated the protein, the Neurocan protein, that are going to be secreted into the medium, and then we're going to analyze them with a Western blot, where we are expecting a high molecular weight band related to the Neurocan. We are expecting 215 kilodaltons. Today, we can conclude that the Neurocan was successfully cloned, and our future directions are to verify the transfection efficiency with a Western blot, and to use the Neurocan as a substrate for the zebrafish brainstem cell culture experiments. I want to acknowledge Dr. Vance Lennon for facilitating the lab equipment in the University of Miami, my mentors, Dr. Tapanes Castillo and Dr. Plunkett, a recent graduate from St. Thomas University, Matt Dodge, also my lab peers, the U.S. Department of Defense, and the NICEF grant. These are my reference. Thank you very much.
Any questions?
How did you go from recognizing the right clones after your gel electrophoresis and obtaining the sequence of the protein?
Okay. We have previous sequences, and we want to, after we have the positive clones, we know that we have the MIG vector that we previously expected. And we want to send them to make sure that because usually the clones might be modified during the process, and we want to make sure that it was actually cloned in the correct way, and that's how we get into the sequencing.
Did you actually cut the band of the gel?
Yeah, we did. We did an extraction band.
And then how do you purify the DNA from the gel itself?
With the, okay, we have a protocol for that. It's a purification extract band. And we also, we heat the gel in order to obtain the Neurocan. And then we do diagnostic gel to see the results.
Can you go back to your first chart, please?
Yeah.
Right. So the ladder, the last one on the ladder is what?
Sorry?
1,000? The last band on your ladder?
This one?
Yeah,
That would be 1.5.
1.5.
Yeah.
You mentioned at some point that you knew that the bands that you were getting are the correct bands because
We
they're
previously,
at the right size.
band size, yeah.
But it seems like the ladder doesn't go that high.
Oh, here?
Right.
Okay.
So how would you know your the size of your DNA?
That, okay, the thing is that we are actually seeing the double digest, and it is higher, you're right, it's higher than the 1.5. So we know that, well, since it's higher than the 1.5, it's approaching to the expected band size.
There's a lambda ladder.
It's a,
There's a lambda ladder right next to it.
Right.
here.
That's the other marker.
Right.
There's two markers on it.
So do you think that it would help by putting the sizes on the lambda ladder
Absolutely.
in your slide?
Yeah.
In other words, to put the relevant sizes and to frame your band of interest with two standards, one above and one below. And that will give you the range.
Yeah.
And also that same slide, up in your diagram, so you have 4 cuts, and there's one cut that is not labeled.
Oh, here?
Yeah, right.
That would be the ZOL1 too.
Also?
The
there are two
ZOL1,
sides?
the restriction enzyme, ZOL1.
So there are 2 sides for ZOL1?
Yeah, we use that ZOL1 first for, well, we actually use another restriction enzyme than the one that I mentioned before, the expat one, because if we use the ZOL1, we couldn't differentiate between the band sizes because they're the same as the Neurocan band. So,
Okay, but you're saying that, you see that there is one side that is not labeled?
this one.
Yes. You're saying that's also a ZOL1 side?
That's an extra, sorry, that's an extra site. So, what she did was in her plasmid map, she shows the ZOL1 site, because that was used for cloning, but that wasn't used for the gel that you're seeing now. The gel that you're seeing now is just EcoR1 and EXPA.
Oh.
The unlabeled one is EXPA, so you see that there's 3 bands. But it